Hello everyone. So in this video we are going to discuss about the 25 top MCQs from DNA. So very first question, DNA stand for. So the full form of DNA is deoxyribonucleic acid because it contains deoxyribose sugar. Okay. So option B is correct. Question number 2, DNA is located mainly in. So DNA कहाँ पे locate होता है cell में? So it is located in the nucleus. Okay. So option B is the right answer. Okay. देन क्वेश्चन नंबर 3, द बेसिक यूनिट ऑफ डीएनए. जैसे प्रोटीन का बेसिक यूनिट अमीनो एसिड होता है, वैसे ही डीएनए एंड आरएनए का बेसिक यूनिट कौन सा होता है? न्यूक्लियोटाइड होता है. ओके, सो ऑप्शन बी इज द करेक्ट आंसर बिकॉज न्यूक्लियोटाइड हैज शुगर, फॉस्फेट ग्रुप एंड नाइट्रोजन बेस. ओके, ऑल थ्री कंबाइन टू फॉर्म ए न्यूक्लियोटाइड. सो न्यूक्लियोटाइड इज द राइट आंसर. Question number 5, which of the following is nitrogen base of DNA? So uracil is the nitrogen base of RNA. Adenine is the right answer. Adenine always pair with thymine in DNA. Okay, ribose is the pentose sugar and glucose is the sugar. It is not the right answer. Question number 5, in DNA adenine pair with. So in DNA adenine always pair with thymine and cytosine always pair with guanine. Okay, adenine has 2 hydrogen bond with thymine. Question number 6, the structure of DNA was discovered by. So the structure of DNA was discovered by Watson and Crick in 1953. They proposed that the DNA has a double helical model structure. Question number 7, the shape of DNA is described as. The shape of DNA is described as a option number B is correct, helical coil like structure. Okay, DNA has a double helical structure. Now come to the question number 8, in DNA the sugar molecule is. So in DNA sugar molecule is deoxyribose. Okay, and in RNA it is ribose. Question number 9, the backbone of DNA is made up of. So the backbone of DNA is always made up of sugar and phosphate. So option A is correct. Sugar phosphate chain form the side of DNA ladder. Question number 10, the complementary base pairing rule in DNA is. So we already discussed about this. The adenine always pair with thymine and guanine always pair with cytosine. Okay, so option C is the correct answer. So the pair with thymine, 2 bond thymine and adenine and guanine and cytosine have 3 hydrogen bonds. Enzyme is responsible for DNA replication. So DNA polymerase is the right answer. Option B is the correct answer because DNA polymerase add nucleotide during replication. Question number 12, DNA replication is. So DNA replication is a semi-conservative process because parent DNA unbind to form the complementary pairs of 2 helix. Okay, each daughter DNA molecule has one old and one new strand of DNA. Question number 13, who proved semi-conservative replication of DNA? So its right answer is A. Michelson and Stell is the right option. Their experiment with isotope proof semi-conservative DNA replication. Question number 14, which base is absent in DNA but present in RNA? Okay, so its right option is C. Uracil is absent in DNA but present in RNA. Question number 15, which of the following bond join the 2 DNA strands together? So 2 DNA strands are joined together by hydrogen bonds. Okay, these hydrogen bonds present between the nitrogen base hold the 2 strands together. Question number 16, distance between 2 successive base pair in DNA is. So the 2 successive base pair, the distance between them is 0.34 nanometer. Okay, between the 2 adjacent base pair. Question number 17, which of the following is not component of DNA? So phosphate group is there, deoxyribose sugar is there, adenine is there, but ribose is not the component of DNA. It is component of RNA. Question number 18, which scientists performed the famous transforming principle experiment? So option C is correct. Griffith discovered that something later found to be DNA could transfer genetic material. Question number 19, Hershey and Chase experiment confirm that. So Hershey and Chase experiment confirm that the DNA is a genetic material. They use bacteriophage label with the radioactive isotopes. Now come to the question number 20, the number of purines equal to number of pyrimidines in DNA. This is called Griffith rule. Chargaff rule. Chargaff rule say that the number of pyrimidines base pair is always equal to the purines. Adenine always equal to thymine and guanosine always equal to cytosine. Question number 21, a DNA molecule is anti-parallel, meaning it means that the both strand run in opposite direction. Option B is correct. Strand run in opposite direction. One strand run 5 prime to 3 prime, and another run from 3 prime to 5 prime. Question number 22, which of the following is a purine? So purine, always remember that प्यूरिन एंड pyrimidine, दो टाइप के बेस पेयर्स होते हैं, प्यूरिन्स एंड pyrimidines. प्यूरिन छोटा नाम है तो इसकी फैमिली में 2 मेंबर ही होंगे, और pyrimidines बड़ा नाम है, लेंदी नाम है, तो इसकी फैमिली में 3 मेंबर्स हैं. अब याद ये रखना है कि प्यूरिन्स में आता है क्या, AG करके आप याद कर लीजिए. AG मतलब एडेनिन एंड ग्वानिन. एंड pyrimidines में आता है कट, CUT कट, साइटोसिन, यूरेसिल एंड थायमिन. सो इसके हिसाब से प्यूरिन्स में आ जाता है एडेनिन. सो प्यूरिन आर लार्जर, 2 ring bases are there, adenine and guanine are the member of purine family. Question number 23, DNA found in mitochondria is. So the option B is correct. The DNA found in mitochondria is circular and double stranded. So mitochondrial DNA is circular like bacterial DNA. Question number 24, the enzyme that unbind the DNA double helix during replication is, it's helicase. So helicase enzyme help to break down the hydrogen bond between the nitrogen bases and separate the 2 strands. Question number 25, the region where DNA replication start is called. So origin of replication is the right answer. Option B is correct. The site of replication is known as ori site, that is origin of replication site. Because replication always begin at the specific sequence called origin.